beginning of a series of calls that we will be hosting for you during this difficult time. To reiterate, our mission doesn't stop, and that is to strengthen bilateral relations between Israel and the member states of the European Union. And I think we understand how important, even in a crisis, even more important, even in a crisis, how necessary it is for heightened cooperation and partnering between Israel and Europe. Just to give you a quick update, while it's not business as usual, we are continuing our program. We have pivoted our program to robust online platforms. Next Monday, we'll be holding another call for our supporters and friends. We will also be having a special call for dignitaries and various officials, updating them on COVID-19. We intend to continue our EU-Israel strategic dialogue. We will do that online. I'd like now to turn over the moderation duties to Felnet President David Siegel. David?
Thank you very much, Lee. And I want to do a special thanks to everyone on this call, our board members, supporters. We're very appreciative. We know that this is a very difficult time for everyone. And as Lee said, we can't stop our work. We're determined to continue. And we are rapidly developing various ways of continuing our engagement with European officials, together with our affiliates in Europe, in our teams on 3 continents, continue the work that we believe is very vital, whether it's the cooperation on the research for a vaccine or for therapies or other medical issues. And certainly Israel's economy, you probably know, and it's reflected in other economies around the world, but there are already over 500,000 unemployed in Israel in a span of a few weeks. We're going to see a significant economic hit that Israel will experience, and Israel will need all the help and all the friends that it has and it can get through its relationships, certainly in Europe, Israel's largest trade partner, largest conduit for exports, imports, but also for funding of scientific innovation and research that Israel needs. So this cooperation is vital. It will continue to be vital. The work that ELNET does and the work that you support will remain critical to our future, both Europeans and certainly Israelis and the Jewish communities in Europe. The work that we have to do against anti-Semitism, where they're also using the virus in attacks on Jewish communities. So we cannot stop, and we can't let travel bans stop our work or closures stop our work, and we're determined to continue. Lee mentioned this. We have a very busy spring, roughly 10 briefings that are already in planning of various stages with our European partners, and of course for our donors, which we hope every few weeks to engage you directly with speakers and experts. So without further ado, we're very, very honored to have Tel Aviv professor Jonathan Gershoni, one of Israel's leading authorities on immunology and virology, who is highly sought after as an expert and speaker on the coronavirus in efforts to educate the Israeli public through the mass media. Professor Gershoni, despite his English, was born in Haifa, and he's a bona fide Israeli. Not only that, but he was the son of one of the founders of Israel's Navy, a Machal volunteer. He grew up in Haifa, graduated from Hebrew University, and completed his postdoctoral work at Yale, and conducted further research at Harvard, NIH in the United States, and the Weizmann Institute in Israel, before joining the Tel Aviv faculty in the early 90s. Now, for the last 15 years, he's been running a lab with major research on the coronavirus, and he is one of the contributors around the world in the search for a vaccine as we speak. So Professor Gershoni, the floor is yours, and let me just ask you, as you begin your remarks, How is this virus different from other viruses that we have faced and that you have researched? And where would you place Israel in its global response to date? And are we at a fateful crossroad?
Thank you. First of all, I must admit this is a bit intimidating, looking at my screen and knowing that you're all listening to me. I hope I have something to contribute. As David had mentioned, I have been working on coronaviruses for the past 15 years. That is in addition to working on HIV since '87, and flu as well, and dengue. So there are a whole slew of viruses we've been looking at. So if this is a virus that we just now discovered, how is it that I've been working on it for 15 years? So to be precise, I've been working on coronaviruses. Humans have 6 different coronaviruses before this most recent event. And to put things into perspective, at least a few of these, 4 of these coronaviruses actually cause rather mild diseases. And in fact, if you will, the common cold, 15 to 20% of all cases of common cold are the result of a coronavirus infection in human beings. So coronaviruses are not new. They carry with them an awe of intimidation and threat, because in 2002, a new coronavirus emerged, which we all learned and began to study, which is SARS coronavirus. It's the severe acute respiratory syndrome virus, and it's particularly threatening and causes a lot of concern because the fatality rate of SARS coronavirus is 10%. To compare it, for example, to seasonal flu, the fatality rate in flu is about 0.1%. So certainly SARS coronavirus from 2002 is a very dangerous virus. However, as it turns out, it isn't necessarily all that infectious. And this is one of the points that we have to recognize, because many of us try to learn from our experience in SARS and remember that SARS actually petered out after about 9 months of infection and about infecting 30 countries. Right now, we're 3 months into the epidemic with close to 400,000 infected individuals in 180 countries, plus minus or so. So this is a very different beast. Now, once the SARS coronavirus, originally from 2002, petered out, we were witness to yet a new SARS virus, or a new coronavirus, Middle Eastern respiratory syndrome virus, which actually is curious because this virus emerged in Saudi Arabia. Fortunately, in Israel, we've never had any cases of MERS. And the animal vector, the reservoir, the natural reservoir of this virus is in camels. So you can imagine the dynamics here in the Middle East, that you have Hajj, you have the holy pilgrimage to Mecca, and you have caravans of camels. And so there was a spread of MERS coronavirus throughout the Middle East. And then there appeared a center of an epidemic in South Korea. And South Korea was very disturbing, of course, to the US that had 80,000 troops there. And so this again was an emerging epidemic. It's far more fatal, so the fatality rate is about 30%. However, it too has not been very infectious. So there are a total of maybe 4,000 cases of MERS. So one can ask, what's the difference between the previous viruses, the 6 coronaviruses that we knew, and this new emerging coronavirus, which emerged, as you all probably know, in Wuhan in late December of 2019. The disease is coronavirus disease, that is to say, COVID, and 19 indicates that it was first detected in 2019. The virus itself is SARS coronavirus 2. And that is because the genetic composition of this virus is very, very similar to the 2002 coronavirus originally discovered. And so you might want to hope that this virus is also going to peter out. It's going to follow in step after its cousin, SARS coronavirus from 2002. And unfortunately, I don't believe that's the case. The virus is very different. First of all, it isn't as lethal as the original SARS. That was 10%. And now we're somewhere in the vicinity of possibly 3% fatality, which is, of course, still markedly higher than flu. The virus is different. It's different in the sense that, first of all, it's devious, it's insidious in the fact that people who are infected, some 30, 40%, have either very, very mild symptoms or no symptoms at all. Now think about it. That's basically putting a population of individuals that are infected, they are carriers, and they themselves don't even know that. Now, that isn't the worst scenario. Actually, the worst scenario exists. Those that have no symptoms are still infectious. They are contagious. They are shedding virus to the environment. So you have a situation here where people get infected. They are passing on the disease without them even knowing that they themselves bear the virus. This is very different than the original SARS. The original SARS was a virus that was infectious only in individuals who had been sufficiently infected and developed symptoms, running nose, a cough, and fever. And we all know from our own human behavior that we have a tendency to separate ourselves and not engage in people who are obviously ill. But in the case of the current virus, we don't have those cues. And so the social distancing that was implemented in 2002, 2003 was far more effective. And so this is one of the major reasons why we saw the original SARS epidemic peter out. Now, there's another aspect which distinguishes between the two viruses, that from 2002 and the current virus. Viruses don't infect every cell of our body. They're very specific. So obviously, as you know, the viruses that cause hepatitis, they are infecting liver cells. And for example, HIV is targeting and infecting our immune cells. And so each virus has the ability to single out target cells. And they do this by the fact that the virus has a spike protein or a membrane-associated protein, an envelope protein of the virus, and that protein of the virus then has to interact specifically with a receptor which is situated on the target cell. So the receptor for both of the SARS coronaviruses happens to be the same protein. However, the affinity, the efficiency by which the old virus bound to its receptor, which is in the respiratory tissues of the lung, was not that great, which means that for the SARS coronavirus of 2002 to be infectious, it had to actually, the infectious dose, the amount of virus that you had to be exposed to was markedly higher, and the efficiency of infection was markedly less. Today's virus, the binding of the virus to its corresponding receptor is very efficient. And so we're seeing not only that we have asymptomatic people shedding the virus, but the virus also has a higher capacity, a greater efficiency to infect individuals and people. And so we're seeing a very different case. We're seeing a rapid infection. We're seeing, unfortunately, epicenters throughout the world, initially in Wuhan, in the Hubei province in China, but certainly and unfortunately, Italy, Spain, the UK is possibly now developing a higher rate of infection, and most definitely the United States. And here in Israel, we have currently 1,200 infected individuals, plus minus. The situation is not good. I would like to be in a position to say that, you know, we are leading in the world with regard to the infectious rate and the burden of the virus in Israel and its effect on the economy. I would say, generally speaking, we're average. We're more or less as other countries in the world, and we're all together needing to collaborate, to interact, to share. This is in contrast, and I certainly don't want to get political, but some of our leaders are indicating that we have a front line, that we are leading the effort in trying to combat this particular virus. I dare say that that isn't quite the picture that I see as a professional, as a scientist. I'm not a physician, and I like to stress that. But as a bona fide scientist working in the field, we're doing a reasonable job. I think we are at a critical position. In the absence of a vaccine, in the absence of drugs, the only real tool we have to try to keep the infectious rate down is social distancing or isolation, quarantine. And this, of course, is a very difficult task, and it affects the whole society. It affects people. Each person is being requested to separate himself from loved ones, family. I've got 10 grandkids and I haven't seen them in a while. We do Skype and Zoom, but that obviously doesn't have the same effect of hugging your grandkids. And so this is certainly a serious problem, and a problem we have to cope with. Will it be effective? Well, I hope so. I think we're at a critical position. We've now implemented social distancing and a emergency state in the country. We've reduced the work task force at various universities. For example, at Tel Aviv University, they've cut the presence and the activity of research down to by 70%. This has direct effect on myself, for that example. I find myself now trying to secure my research staff so that we can proceed and do the necessary work to develop a vaccine. So, you know, it's really difficult times in that respect. And so you can ask yourself, so what are the solutions? The immediate solution as far as national policies in Israel, in Europe, in the United States is social distancing, isolations, quarantine. Of course, everybody has talked about humming happy birthday to you as you wash your hands. Certainly, without a doubt, hand and hygiene, good warm soap and water is definitely very important. We all have the tendency to touch surfaces and then scratch our ears, put our fingers in our mouth, rub our eyes, or hold a sandwich. All of these are efficient ways of transmitting the disease. You pick or you touch a contaminated surface, and then you pick up a sandwich, and you may be in a situation where unknowingly you've transmitted the disease and you've contracted the disease. So hygiene is important, social distancing. So then, of course, the question becomes, how long are we going to have to be stuck at home? Now, for some of us, being stuck at home ain't bad, but most of us would like to get out there and carry on and get on with our lives. And with that in mind, the real effort has to be, in addition to the social efforts, is to develop a vaccine first and foremost, and to try to identify, discover, and implement various medications. For example, I think you've all heard about the fact that the company, for example, Gilead, has been testing various drugs with the hope that they will be found effective in treating COVID-19. Now, why is that advantageous? Well, these companies, and Gilead is definitely a remarkable company. I'm not pushing it here, so I wouldn't go out and immediately buy stock. However, on the other hand, they have produced a whole panel, a collection of very, very effective antivirals. Now, you have to remember, virals are not bacteria, and so antibiotics aren't going to work. But the cocktail, for example, for HIV has been markedly improved by Gilead, and its effects in managing the AIDS epidemic has been remarkable. Curing hepatitis type C is actually a reality, and that is the result of developing very effective antiviral drugs. One of the drugs that Gilead's has developed was a drug they developed to try to curb and cure the effects of Ebola. It went through phase 1, phase 2, and phase 3 trials. Unfortunately, it was not found to be effective. However, that gives us an opportunity now to test its efficacy against COVID-19 disease without having to go through all the regulatory hoops and obstacles, because it's already been approved as far as the regulatory aspects. So here's where this could be definitely an advantage. Unfortunately, in the recent reports on trying to implement and use HIV-directed drugs, which have all been approved, they weren't found to be efficacious or able to actually improve the health of those who are suffering from COVID-19. You've also heard, of course, of other candidate drugs such as chloroquine and others that people are grasping. People are trying to take out of their toolbox various drugs that have proven to be effective to some diseases and hope that they may also have some therapeutic effect in this current epidemic. So this is going to be ongoing. Another thing that certainly will be done, however, this will be more time consuming, is the development of new drugs. When we develop drugs for a virus, we have to try to seek some sort of viral component, some sort of an enzyme that is unique to the virus and very different from our own enzymes. And then you can possibly find something to inhibit that enzyme and gain what we refer to as therapeutic index, the ability to kill the virus without necessarily causing too much damage to ourselves. And so there will certainly be now an aggressive effort to try to identify new antiviral drugs targeting specifically the SARS coronavirus 2 and trying to be tested for the efficacy in treating COVID-19. So this, of course, is something that's being done. Another aspect, which is something to do with what we're doing at Tel Aviv University, is trying to develop vaccine modalities. Now, this is very complicated. And unfortunately, even if we're optimistic and believe we may have a unique approach that could lead to an effective prophylactic, a preventive vaccine, this is going to take at least a year, if not 2 years. You have to think about it, that in the process of developing a vaccine, not only do you have to discover the component of the virus that you want to inject so as to mount a protective immune response in the vaccinee, but then you have to go through phase 1, phase 2, phase 3 clinical trials, licensing, and then you have the enormous task of actually manufacturing, let's say, 500 million doses to vaccinate the world. So this cannot be done overnight. So unfortunately, in Israel and around the world, we're going to have to figure out how to maintain an economy on the one hand, and nonetheless ensure and implement some level, and currently a very severe level, of social distancing. So that's approximately what I can say about the disease. Now, what about our European allies and collaborations with Europe and the United States? This is exceedingly important. This is something that in contrast to some of the flavor of the reports of our prime minister, and as I said, I will not get into the politics, but Netanyahu has a tendency of constantly trying to push how Israel is leading in the effort, and we are the startup nation, and we will have the solutions. ki mitzion titzeh Torah, you know, that we're going to be the source of the solution. Well, I wish and hope and will applaud him if he is correct. However, the reality is the world globally, Europe, the United States, of course, Asia, Africa, all continents were confronted with a very severe and global epidemic. And what we need to do is to work together. This is not a competition. And so I can say from my own personal work, I have colleagues in the United States, in Europe. Quentin Sattentow is a good friend from Oxford, and we've collaborated over the years. And Luigi and Franco Buonagono, who are in Italy with Paolo Lusso. There are a lot of scientists, and at least the scientific community, we tend to interact, and I'm at least encouraged and find it gratifying that at least amongst the scientific community, we all realize that we have to work together. I just received some critical reagents from Neil King in University of Washington in Seattle. And so we're discussing how can we possibly develop effective solutions, vaccines, therapeutics, and diagnostics. And so as you realize it, and I implore that we have to find whatever ways we can to expand and deepen our networking and our ability to support research in each of your countries and in Israel, because we have an ongoing dialogue. And this, of course, is critical to the success which we're all looking forward to in the near future.
Thank you, Professor Gershoni. Gary Lazar, you had a question, I see.
Yes, thanks very much for the great update. Recognizing that there are tens of millions, if not more, worldwide of people who are on ACE inhibitors, and there is conflicting data coming out about the risk of ACE inhibitors in terms of increasing the receptor. Can you make some comments about that?
You surprise me. It's a great question. It's obviously one of somebody who is equally knowledgeable to me, so I might have to seek your advice on things. ACE is the receptor for the coronavirus, and ACE is also a natural protein, which is critical and important in regulating, let's say, blood pressure. And so therefore, the question is are we going to find any possibility of using the knowledge about ACE as a potential therapeutic target? There are a number of ways of going about this, and I'm going to step back to an approach that we promoted many, many years ago, and that is to try to develop various mimetics of the receptor so as to be able to sequester the virus. So at least in my perspective, I think that the new drugs that we're going to have to use are going to have to focus on the viral proteins, on the spike, on the envelope protein, those aspects of the spike that interact with ACE, with ACE. And I would not expect, exactly for the reasons that you said, that we're going to see a lot of drugs targeting ACE as a therapeutic for COVID-19. And I think that's a very important point you pulled out.
Thank you, Gary. We have a question from Germany, from Eugenia Levine. Is there no licensed late-stage clinically tested vaccine that could be repurposed similar to what is happening with treatments like remdesivir?
Okay, this, again, if we're referring to a vaccine, there are about 30 different industrial and academic efforts currently, and I'm sure there are more, trying to develop a vaccine for SARS coronavirus 2. As I mentioned, the two previous SARS coronaviruses that were threatening the world, the SARS coronavirus 1 and MERS, there have been serious efforts in trying to develop a vaccine. And unfortunately, they have all failed. So the general model for the vaccine for these viruses has been to try to generate a recombinant, the product of biotechnology, of the envelope, and then use that as your vaccine, hoping to raise antibodies that will target the viral envelope and therefore ultimately inactivate the virus should we get infected. Now, this is a very valid and quite honestly a very good approach. And this is precisely the approach which is used in generating Gardasil, the vaccine for human papilloma and cervical cancer, and as well as the vaccine for hepatitis B, which is given to kids almost at birth. And the concept is not to inject a virus, an attenuated or a killed virus, but just the viral envelope protein. Unfortunately, however, all attempts thus far to develop an effective vaccine for SARS and for MERS have been attempted but failed. So to reuse and try to relabel a successful vaccine would be a great idea. Now, the closest thing to that, which you've mentioned, is that we do know that there are a number of very effective antibodies, monoclonal antibodies, that do knock out both MERS and SARS. And the question immediately was, can we use pre-existing antibodies to the cousin viruses and test to see how effective they might be in knocking out the current virus? And unfortunately, whereas there is genetic similarity, there's sufficient dissimilarity and difference. And therefore, those antibodies, as effective as they may be for the other coronaviruses, have not proven thus far to be effective for the current virus. I hope that answers the question.
Thank you. A question from Michael Levy. The world powers are accusing themselves in this period, and there are many questions. What is the real difference between a lab-engineered coronavirus and a natural virus? How is it possible to understand and exclude that SARS coronavirus 2 did not spill out of a lab?
I'm debating how to respond. Basically, I can say it, that this is total nonsense. Not the question. The question is, how do I make sure that it isn't? But the conspiracy theory that outside some lab in Wuhan, SARS coronavirus 2 was generated as a potential bio threat of sorts and escaped from the lab, and therefore we're confronted with a humanly engineered virus. This is good for a movie. I remember in 1976, when I was a young molecular biologist, there was the Cambridge City Council that actually banned all research on recombinant DNA, people being concerned that scientists are going to create some threatening monster bacteria. And so it's taken years, in essence, to gain the confidence of people and the populations, the scientists and the politicians, that this doesn't happen. Now, I'm not saying that somebody could not possibly generate a devious biological threat. I think that if I were to do it, and don't quote me on this, I'd probably pick a better virus or a better bacterium. But in any case, in the case of what is being proposed for SARS coronavirus 2, is that there was a single paper that I came across in which a scientist did compare the genetic composition of the current virus and ask, are there fragments of this virus that might have been picked up and possibly incorporated from other viruses? And so, of course, the first test that he looked for was, are there any segments of HIV, the AIDS virus? And doing a computational analysis, which could be no more than mediocre, he found very short segments, 4 segments, that he claims must have been the product of genetic engineering to incorporate them. Now, because it's my training to actually critically evaluate what people are saying and take them seriously and give them the credit, we made a very systematic and comprehensive analysis of exactly what is being claimed. And one of the major points that needs to be stated is that these same 4 short segments could also be found in various coronaviruses of bats. And we believe that the current coronavirus is in fact probably derived from bats, as are many coronaviruses. Moreover, the fragments that he is suggesting somebody engineered into the coronavirus don't make sense. Although he claims that at least 2 or 3 of the segments are associated with the viral envelope of HIV, and therefore he argues this might make it a super virus. I've been working for the past 30 years, actually from 87, on HIV envelope, in addition to working on SARS and Corona. It doesn't make sense to one who is a critical scientist. The problem is, however, that in today's Facebook, Wikipedia, accessibility to information, the thrill of a conspiracy, and the lack of rigor of those who are promoting the conspiracy theories, it makes a lot of noise. If you were dared to ask any one of the people who promote these concepts and these ideas as to, well, did you read the article? And if you did you understand the article? And did you critically evaluate the data on your own? I would be surprised if you'll find more than 0.1% of all those who are promoting the idea that actually did the due diligence. And I use 0.1% because that was the fatality rate of flu that I quoted previously. So I don't believe it. I will be the first, as we say in Hebrew, to eat my hat if it turns out that I'm wrong. But I think it's total nonsense.
Thank you. We have a very engaged group here, so there are many questions in the pipeline.
I will try to shorten my answers.
Well, I'll try to give you short questions. So from Marsha Gilbert, what about hydrochloroquine as a therapy? And then a question from Daniela Oliel about the questions about ibuprofen and does it encourage the spreading of the disease?
I can answer both very quickly. I don't know. However, chloroquine has been proposed as being potentially helpful. I have no idea. And Advil and similar drugs have been argued that might be deleterious, and I simply do not know. That isn't to say that I would be concerned, but I just don't know.
So sounds promising, at least one of them. So we have a question from Mr. Azenstein. Am I saying it right? What is the most likely continuation of this pandemic? And is there a ballpark in terms of a possible timeline? This is from Daniel Azenstein.
Daniel, I tried to address that initially. We're not in the position that we're going to see it peter out like the previous SARS, which was in the period of 9 months, mind you, and we're only 3 months in. I'm very concerned that we're going to see yet a peak in the future. I don't believe that we're beyond the peak. And then in view of the fact that it's an effective virus in its infectious rate, and we don't have a vaccine yet, I think we're in store for more months to come. There's one point that I want to make, which is relevant. People are raising their hopes that as we come closer to spring and summer and higher temperatures, this might be deleterious to the virus and ultimately cause it to be subdued. But for those of us who may remember when in the 50s polio was a threat, polio season was summer. So I don't know. I will be delighted to see that there's a drop in the infectious rate as we enter spring, but there's no indication as yet of that.
Daniel, thank you for that question. We have a juicier one from Arya Ben Simon, who is our director in France. Is the Israeli government violating individual rights and freedoms through the technological tools it is using to monitor the epidemic? A very important question.
Aria, bonsoir. In reality, I personally, I'm confused because I'm looking at the camera, but I want to look at you too, and you're down on the screen. So I'm going to continue to look at the camera. First of all, it's curious because a number of applications have come out that you can voluntarily hook up to, so it does, for the most part, the same thing. There certainly is a level of violating personal privacy. And I would agree that this is definitely a serious question for debate. The proof of the pudding is going to be in the eating. I know of individuals here at home in Israel that have received messages over their phone. You are now having to quarantine yourself. You were in the vicinity on a particular day to somebody who has now been diagnosed as being positive. So if in the end, and I think that there is a chance to actually measure the efficacy of this policy, if it turns out to have been truly a critical tool that was able to allow us to curb the rate and flatten the curve, then some of us who are concerned may feel a little better. If in the event the measurement and the measures, the measurables of its effect turn out to be negligible, then again, it is a spin and unnecessary. I don't see that it's been used in other countries. And so we're all concerned. And if on the other hand, it turns out to be extremely effective, then I think we will all in hindsight feel that it was justified.
Thank you. We have a question from Bradley Schmarak. It seems to me that we need to get people who have been infected by the new coronavirus and who have recovered back into the global economy. How can we test people to determine if they have antibodies and can get back to work without the risk of infection to themselves and others?
It's a good point. It sounds reasonable. However, it's a bit premature to actually be confident that it's a valid approach. People, when they become infected, it is true that they mount an antibody response, and we are hoping that this antibody response will be protective and prevent second rounds of infection and also be long-lasting. However, the current situation is such, we don't have enough experience, and we don't therefore know if the antibodies that are actually generated, they do in fact kill the virus efficiently and prevent infection. So it would be horrific if we found that such antibodies were proposed to provide protection, but ultimately did not. So now you've actually said to people, you've already had one round of infection, you can now go out because we're confident that you're protected, but if they are not, that could be dangerous. I don't want to raise a red flag. However, there are diseases that actually, in the second bout of the disease, it is more devastating, and that is because there is a phenomenon called antibody-dependent enhancement, ADE. Now, I don't believe this is the case in corona, so don't quote me on the fact that I'm concerned about ADE in corona, but I'm just saying that things are very complicated. We know, for example, that people who become infected with HIV, they mount an antibody response, and in fact, it is precisely these antibodies which are the basis of an AIDS test. But these people who have mounted the effect are not, by their antibodies, becoming cured. They're not surviving the disease. So this is still up in the air. We don't know if the antibodies that are generated during the course of infection in fighting the disease and recovery, are they going to be sufficiently protective? And we don't know if it's going to be long lasting. And then there's a last point to this general hypothesis. The people who are infected is a random selection of our population. And if we look here in Israel, because this has been proposed in Israel as well, we have about 1,200, maybe 300 infected individuals in a population of 9 million or so, and let's call it 4 or 5 million people as our workforce, let's assume. But of the 1,300 people, you have a whole slew of elderly people like me, and then you have younger kids and students, and certainly the remaining people are a spectrum of all professions, a profile of all professions in Israel. So they are not the workforce that's going to salvage and really save our economy. So generally speaking, you would hope that antibodies are going to be effective. In fact, that's the basis for vaccination, to mount antibodies in the vaccinated people sufficiently so that they are protected. And this is what everybody's working on. But to assume that antibodies are going to provide and rejuvenate the workforce, I think that is probably not the case. It may contribute somewhat, but not totally. A better use of antibodies is to try to draw blood from people who've been infected and to purify from them antibodies that are proven to knock out the virus, and then use those antibodies as an immunotherapeutic passive vaccine. Somebody who's in a critical medical situation, to give him a dose of antibodies derived from people who have recovered. That's very valid.
Thank you, Professor. We have a question, another question from Germany, and I just want to make special mention of the group here from Germany today. This is from Klaus Thormellen. I apologize if I pronounce the name not accurately, but he's asking a very similar question about the immunity of those who emerge from the disease, and if so, how long will they be immune? So maybe we can use this opportunity to talk about the antibodies for a moment and the potential for European-Israeli cooperation, since Israel has such a small pool and Europe has a much larger pool. Is this an area where we can cooperate with Europe in an effective way?
I think so. Guten Abend. I'm glad for the question. And frankly, we have here as I just mentioned, in fact, an application. Let us assume that we are able to develop a good target for antibodies. We would like to use the target as a vaccine, but intermittently, we can try to find immediately relief and a potential therapeutic for the critically ill. And that would be by collecting blood from recovered people and their serum, and then trying to purify their antibodies and using them as therapeutics. So this is a very valid approach. There are companies in Israel that actually do this and have created various drugs, let's say for West Nile disease, which is another type of virus. And so here we need, I think, the support of Europe. And it's somewhat cynical, possibly, it sounds that way, but that isn't my intention. Europe, Italy, Spain, the UK, Norway, other countries have been hit and have far more individuals that are infected, and they can be a source of blood that could be used to manufacture or at least isolate and produce good, effective antiviral therapy, and to be used predominantly in the more critically ill. So I think that this is an area where we can interact and see if we can help each other develop good therapeutic reagents.
We have a question from our very own Pascaline in our team in Israel. And she would like to know what the world can do to prevent the next pandemic. Our strategic folks are always thinking one step ahead.
Hi, Pascaline. How are you doing? It's good to hear from you. Another world network, which I am a member of, is called the Global Virus Network, the GVN. And the GVN was initiated a number of years ago, I would say well over a decade, by a group from Germany, from Ireland, and from the United States, Bob Gallo. And the idea was to create a network of virologists that will do precisely what you're suggesting. That is to say, be prepared for the next epidemic, the next threat. We communicate amongst ourselves, as I said, and I'm stressing that developing, expanding, and deepening networking, just like in ELNet, is exceedingly important for success. I would say most certainly that we need to be prepared. I don't want to get into it right now because it's still a bit under wraps, but the vaccine that we're trying to develop is a patented technology that we filed for patent in 2015. So this is exactly what we need to do. We need to be able to learn the details of the existing threats and think about how to project and use that knowledge in order to counteract and intercept threats for the future. And so I think that the dialogue, the collaboration between laboratories in different countries to have debate, discussion, sharing information, strategies, and technologies. I had a group from Russia come and learn about the technologies that we've developed. And we've just had, in fact, 2 Kenyan scientists working in my lab to learn about the technology and technology transfer so as to do precisely as you're suggesting. Share as much as possible and try to anticipate how this information can be implemented in future threats.
Last question, I think it's a question on all of our minds. This is from Richard Hoffman. If one is quarantined for 14 days without symptoms, is one reasonably assured that they are not infected? And what are your thoughts on herd inoculation?
And you want me to answer that in a minute and a half?
Yes.
The reality is this. As I mentioned from the very beginning, one of the major problems of this virus is that it can be infectious in the absence of symptoms. So for the general assumption that if you were to actually develop the disease, you will show symptoms within the course of 14 days, and therefore 14-day quarantine makes sense. On average, that's a pretty good guesstimate. In reality, it's not 100% assurance, because there could be the situation that a person was infected. He ultimately actually developed the asymptomatic disease. He then subsequently feels as good as he was when he was asymptomatic. He doesn't seem to sense, but this virus is devious in the sense that it tends to linger on. If you look at the numbers, you'll see that there are a lot of people who are active cases and very few that are recovered, at least in Europe and in Israel most definitely. And the reason for this is what we define as being cured, as being recovered. And the definition is that you are tested at least twice consecutively, and you have to come out PCR or diagnostic test negative for the virus. And it turns out that a lot of people who have been infected and feel great have not been able to effectively clear the virus for many weeks. And in fact, it was just 2 days ago that a woman from the Diamond Princess was finally released. She's been feeling pretty good, but she was actually quarantined for 6 weeks because she constantly had a lingering amount of virus. Now, we don't know for sure that this low dose of virus is equally infectious, but it could be. So there are no promises here. As to herd immunity, That is precisely the objective of a good vaccine modality.
We really want to say from the bottom of our hearts, all of the LNet family, you've all just participated in our really experimental forum, the first time that we've done an online engagement like this. And what this means to us is there is a silver lining, because we could expand our networks that much more online and reach more countries and more officials without necessarily having to always physically be on the ground. So there's tremendous potential here for the continuation of our work. And we call on all of you to join us in this effort. We deeply, deeply believe that we're fighting an invisible enemy. And this means that economies will be absolutely devastated, which means that Israel and Europe will need each other more than ever before. So that the work that our mission will only be more important than before. And we thank you for believing in us and continuing to invest in this effort. Because in the end, what we need are real leaders that are dedicated to a greater cause. And this is a very, very important cause. So Professor Gershoni,
I would like to
thank
say
you
one
very
last
much.
word.
Sorry?
Can I just say one last word?
Absolutely.
Yes. So first of all, Larry, I want to thank you very, very much. I'm thrilled, I'm honored, and humbled by the effort that LNet is making in putting this together, in demonstrating the efficacy and how wonderful it is, the ability to network, exchange, and share. And I applaud you all. Thank you very, very much.
Hi, I'd just like to say one thing to everybody. If you did benefit from today's call, we will be having another one this coming Monday, a week from today at the same time, 11 a.m. Eastern, where we will be hosting former Brigadier General Michael Herzog and Friends of LNet Strategic Advisor Steve Rosen. We will be talking about the coronavirus and its effects on Mideast geopolitics. We think it's a very important subject, and we hope you'll join us. We will be sending out invitations shortly.
Thank you, Lee, and thank you all for joining us.